Hi, and welcome to my report about the deep sea ecosystem. So I hope this topic find you interested in this vast and least explored habitat on Earth, so the deep sea. Okay, so included in this report actually are the 11 major topics from the very historical discovery of the deep sea up to the huge interest of humanity to explore this very dark and dangerous ecosystem at the very bottom of the oceans. So don't worry about too much information that need to be absorbed here because I have, in fact, provided a comprehensive, detailed dish report through handouts, in PDF form. So, and I'm going to share that to you via the Google Class as well. All right. So, as an introduction to the deep sea, so deep sea is by far the largest ecosystem on our planet that comprise about 90% of the volume of the oceans and with a depth between 200 meters to uh, or and 11,000 meters below sea level. So it is estimated that about 50% of the surface of the ocean or the surface of the Earth is in fact covered by the ocean that is more than 3,000 meters deep. So the HMS Challenger in 1872 to 1876 marked the beginning of what they call the heroic age of the deep sea exploration. And since then, our knowledge has also progressed with the technological advancements. So the deep sea is a part of the marine environment below the photic zone. So this in the photic zone is an area in the ocean where there's no or there is a photosynthesis occurs, and actually it comprise the dysphotic or what they call the twilight zone in the apodic zone or the zone of permanent darkness. So these ecosystems are actually characterized by different ecological factors such as the absence of light, the intense and increasing pressures with depth, and also the extremely low temperature, uh, with few exceptions. For example, in hydrothermal vents, and of course the scarcity of food. So there are many sampling methods that are being used by the scientists to study and explore the deep sea environment, and that includes high definition seafloor mapping using sonar, also using manual operated submersible vehicles, also ROV or remotely operated vehicle, and AUV or what I call it autonomous underwater vehicle. Also deep towed vehicles and sampling equipment, landers as well, um, hydroacoustic instrument, isothermal and isobaric chambers, and finally the molecular technique in laboratory settings. So today, the deep sea, uh, fishing, mining, exploration, oil and gas extraction are just few of the um, factors that are now rapidly affecting the sea environment. All right, the exploration of the deep sea began in the 19th century when Edward Forbes from the Edinburgh University joined the dredging exploration in the Aegean Sea. And he actually observed that as the depth increased, the number of animals decreases. So he thought the ocean below 600 meters could not support life. So this observation of Ford became the azoic theory. And then in 1850, the Norwegian Michael Sars published the list of animals he noted below the 550 meters down or depth with 19 species. And then his son, Ocean Sars, extended the list to 92 species the same year. And then between 1872 to 1876, the expedition of HMS Challenger, led by Charles Thomson, demonstrated that life was actually present in the ocean. And when we say present, he found that the ocean from the shore to the very depth parts of it, teeming with life. So, in fact, it was the Challenger expedition that was considered by many scientists to be the true origin of the modern oceanography. So, the Bibb bathysphere in 1930 was the first deep water vehicle that ventured the deep seabed. And then the deepest surveyed point of all the Earth's ocean can be found in the Challenger Dip, or what we also call the Great Depression. So it was named after the HMS Challenger, where the first recording of its depth was recorded. And then in 1960, Wallace and Piccard explored the bottom of the Challenger Dip using the Trusty Bodyscape vehicle. And actually, they found a small flounder-like fish swimming at the bottom of the Challenger Dip. Okay, so in less than 30 years ago, in 1977, the hydrothermal vents were discovered in the Galapagos Reef in the Pacific. And they found out that the black smokers are colonized by a dense population of exotic giant tube worm. To study the ecosystem or the deep sea ecosystem, so marine biologists requires a platforms, and this could be in the form of research vessels or submersible vehicles. So there are different methodologies to evaluate the biodiversity of deep sea organisms. So the first one is a sonar sampling. So scientists use a sonar like CTDs to determine first the geophysical characteristic of the oceans or the habitat of the marine organisms that live in there. So marine biologists is really interested into that. So the CTDs can be able to measure, for example, the conductivity, temperature, and depth using the sonar signal. So the picture on the right here is an example of a deep-towed side-scar sonar, or CTD, that can be able to produce an acoustic image of the seafloor with detailed geophysical information at the bottom of the ocean. All right, so conventionally, the most widespread method to sample zooplankton in the deep ocean is simply using nets. And usually it is being towed in a research vessel. I guess a great example of this is or are the MOCNES, or the multiple opening closing net environmental sensing system, and I don't know, the thing they called it, the multi-net. And probably the common fish sampling method that is by trawling, which could be either in the water column or along the ocean floor. Also, to assess the benthic organism in deep sea, so marine biologists uses sledges or dredges, box scores like the one at the bottom picture on the right here. So what they do with the box score is that they take sediment samples from the deep sea, and from there they could be able to extract information about the biological and physiological properties of the benthic community in the deep sea. Perhaps the most sophisticated method to study the ocean or the deep oceans is by means of landers or ROVs, or the remotely or remote operated vehicles, like those submersible vehicles here that you can see in the picture on the right. Okay, so these underwater technologies can be able to take massive information when used in combination with the other sampling methods like sonars, because CTDs, for example, can measure the hydrological parameters of the deep sea, and at the same time, these advanced vehicles may study the organisms in there. And lastly, the laboratory or marine molecular technique can be able to study the biodiversity of the deep sea on a species level. So usually they got the animals from there and then process them in the lab to extract information. This is some sort of very important for medical and biotechnological field, because some of the species at the bottom of the ocean are able to give or produce compounds that are important in the medicine field. Okay, so I have here a flashed some of the human occupied submersible and remotely operated vehicle lists that are currently used for research in the chemosynthetic ecosystem at the deep sea environment. And as you can see, the countries like France, Germany, Japan, USA, and UK are the current leaders in exploring the very depth of the ocean. Okay, so there are, or I have here research of 5 key environmental characteristics of the deep sea. So the light, salinity, pressure, oxygen, and temperature. And in the later part of my report, we will dive into this or how these environmental factors actually affect the organism that lives that lives in deep sea. Okay, so natural light does not penetrate in the deep sea, but with an exception in the upper portion of the mesopelagic zone. So this is near the euphotic zone. So the only 2 natural sources of light in the deep sea are actually the sun, the moon, and stars, and the other one is through the bioluminescence light. Another environmental characteristic of the deep sea is the pressure. So the pressure in the deep sea environment increases in every 10 meters. So for example, at the depth of 5,000, that's 5,000 divided by 10, so it is 500 atmosphere, or 500 times that of the pressure of the in the surface of the water. Okay, so salinity in the deep sea environment is pretty much the same as the other parts of the ocean. So it's in the average of 35 parts per thousand, or 3.5%. Also, oxygen in the deep sea is very less to extremely low. So below the 2,000 meter depth, the concentration of oxygen in the water decreases all the way to the bottom. And in parts of the ocean where oxygen is on its critical or lowest level, so marine biologists call these zones as the oxygen minimum zones, or OMZs. Okay, so the 5th environmental characteristic in the deep sea environment is the temperature. And in the upper 100 to 200 meters, or meter depth of the ocean, or what they call the mixed layer, the temperature is pretty much constant. And then suddenly a rapid decline in temperature increase in depth. And this is what we call the thermocline. So about 90% of the total volume of the ocean is actually found below the thermocline. And from there, the average temperature is now between 0 to 3 degrees Celsius. You can see it from the bottom picture, and the pink colored portion, or what I call it, the water where the temperature ranges from 0 to 3 degrees is actually within the mesopelagic zone all the way to the bottom of the ocean. Okay, just a quick review of the physiography of the deep sea environment. So oceanographers divide the majority of the ocean midwater into 5 distinct zones. So these are epipelagic, mesopelagic, bathypelagic, abyssopelagic, and the hadal zone. I actually excluded the epipelagic in this report because technically this zone is not actually included in the description about the deep sea. So it is just the meso, bathy, abyssopelagic, and the hadal zones that I'm going to give a quick run. Okay, so the mesopelagic zone is the zone between the depths of 200 meters to 1,000 meters. So the lights here are barely filtered or filters down, but many animals thrive in here. For example, about 90% of the world fish, like the bristlemouth in that picture, live in this part of the ocean. Okay, so the mesopelagic zone contains the thermocline. And as I have said earlier, this is the transition layer of the temperature change. So at the bottom picture, at the bottom right picture here, you can see that the thermoclines between the depths of 200 meter to 1000 meter around this zone. So the sonar operator of a Navy vessel while sailing during the World War II were actually puzzled by the sudden rise and fall of the seafloor between 300 to 500 meters during the day, and it gets lesser and lesser at night. Okay, so they were actually really baffled by the quick changes of the ocean floors. So wondering why the seafloor moved that fast. But later they found that it is actually a false seafloor which was caused by a diel vertical migration of the mesopelagic fishes. So a large number of fishes were doing the nightly migration to the surface of the water in search for food. So later they call this layer as the dip scattering layer, or a DSL, because it actually does scatter the signal of the sonars. All right, so the bathypelagic zone is the uh, a deep sea area between 1,000 to 4,000 meters beneath the surface of the oceans. So the light here is totally nothing or apophing. So it has a void light, or it is an area with void of light. The temperature here falls below 40 degrees Celsius. So the pressure in the bathypelagic zone is so intense, so at an average of 110 times that of the pressure of the surface. And because of the extreme coldness here, and intense pressure and lack of light, so the organisms that live in this zone must live with minimal food, and that causes slow metabolism for the animals that live in there. For example, in this anglerfish in here, the right side picture. All right, so the abyssopelagic zone extends from 4,000 to 6,000 meters down to the seafloor, or what they call the abyssal plain. So it is the largest ecosystem that covers about 300 million square kilometers wide. Also, it is estimated that the abyssopelagic zone, or the abyssal plain, covers to about 60% of the global surface, and that covers an area of the oceans and seas as well, so approximately 83%. Abyssopelagic zone is very dark and extremely cold, with an average temperature of 2 degrees Celsius at 4,000 meters deep. So there is no photosynthetic activity happening in this area, so oxygen replenishment is not actually possible. Therefore, creatures that live in there must or only consume oxygen of what it is provided. So the abyssal plain, or the seafloor below 4,000 meters deep, is mainly composed of calcareous foraminiferous zooplankton like Coccolithophores, Lephurus. Also, the shells in the abyssal plain tend to dissolve due to the extreme concentration of carbon dioxide in the water. And finally, the sediment here is mainly characterized by clays and siliceous remains of regilargans and diatoms. And finally, the Hadal pelagic zone is the deepest part of the ocean. So this one extends 6,000 meter to about 10,994 meter. And that includes the deepest trenches, like the Mariana Trench. Just recently in 2018, Dr. Geringer found a snailfish at 8,000 below the Mariana Trench. So this is by far the deepest living fish ever found in our oceans. Although there is another fish species that captured recently in a video, but this one has yet to be captured, so it has to be come formally described in science. So the Hadal zone is a very extreme environment because of the absence of natural light in this area, and also the low availability of foods and so much intense hydrostatic pressure. Okay, so based on my research, I have grouped the deep sea habitats into two, and that's how the foods are available in the ecosystems. Although both ecosystems tend to overlap to each other. So the first one is the heterotrophic ecosystem, and the other one is the chemosynthetic ecosystem. So the first heterotrophic ecosystem that I'm going to discuss here is the continent or are the continental margins. So this habitat covers the 30% of the world's seafloor and the largest contributor of the sediment on Earth, with up to 90% of the sediments are generated by soil erosions or on the margins of it. So the continental margins have two types, so a passive margins and an active margin. The passive margin one, or the passive one, occurs when the reef of the ocean is split into two, and that creates a basin in between. So in this photo here is an example of a passive continental margin. So the active margin or the active margins are created when a tense seafloor sinks back to Earth. So for example, deep sea trenches along the subduction zones are considered a active margin. The diversity of faunal biodiversity in the continental margins depends on the spatial scale. It could be either a large one or a mid-scale one. So a super large continental margin of more than 1,000 kilometers, for example, the biodiversity is being influenced by the geology of the area, the temperature, current, and water masses. While on a mid-scale, the major known factors identified are a downflux of a primary production, oxygen concentration, the type of sediments, and also catastrophic events like cyclones. So in this region, the animals are sediment feeders in deeper areas, and the megafauna and suspension feeders increase with the currents along the shallow waters. Okay, so underwater canyons is another type of a heterotrophic ecosystem in the deep sea. So this habitat is characterized by deep incision or step-side walls of the continental margin. The major contributor to the erosion of the underwater canyons are turbidity currents and underwater landslide. So today, about 9,477 canyons are described, and that comprise 11% of the continental margins. So this picture here is a 3D view of the canyon in the offshore of Princess Charlotte Bay in Australia. And as you can see, those step walls forming the edge of the continental margins. This ones. So they are known as underwater canyons. Just imagine the Grand Canyon in the USA. So this is pretty much similar to that. Okay, so underwater canyons act like traps or conduit of sediments and organic matter. And they are considered biodiversity hotspots, particularly for cetacean species. Okay, so the deep water corals, or also known as cold water corals, are the deep sea coral version of the tropical corals that we usually found near the surface of the water. So this one extends beyond the um, 4,000 meters deep with the average temperature of 40 degrees Celsius. So the two photos here are the types of water corals found in the deep sea environment. Okay, so the deep sea coral do not require sunlight to do the photosynthesis, but instead they utilize microscopic organisms like zooplankton, crustaceans, and even krill as their source of foods. And they don't have nematocyte to stun their prey, but they have they have no zooxanthellae as well to feed them. So they rely on the food that they trap, or the prey that they trap, like the small crustaceans and the zooplankton. That is why, that is the reason why they tend to grow more slowly than the corals in the photic zones. Okay, so today the deep water corals are important in the biotechnological potential. That's what I'm saying before, because the scientists were able to extract certain compounds from this organism to synthesize certain molecules, for example. So that, this is very important, or has a medical importance in the biotechnological field. All right, so seamounts are another example of a heterotrophic ecosystem in the deep sea environment. They are usually characterized by steep slope with a hard and substrata. So currently, there are about 100,000 of seamounts in the oceans, and mostly are above 100 uh, um, 100 1,000 meters high, and one of the examples here, as in the right photo, showing us the topography study of the seamount of Kaunana, that's in Hawaii. Okay, so biologically speaking, seamounts have high productivity and mostly large stocks of commercially valuable fishes coming from there. Also, they have high biodiversity with a very high degree of endemism for benthic fauna that can be found in there. Okay, so I have mentioned earlier anoxic area are mid-water oxygen minima that I can actually intercept with the continental margins. And the result sediments will lead to the very low concentration in the water. So these areas are called OMZs, or oxygen minimum zones. So OMZs are typically found between 200 meter and 100 meter in the or 1000 meter in the Eastern Pacific region or in the Northwest Pacific region. Also, they can be found in the Philippine Seas, so the Bay of Bengal, Arabian Sea, and the southwestern Africa. with a very low concentration of oxygen in the water. So the major fungal groups in these areas are foraminiferans, nematodes, ciliates, flagellates, also polychaetes, gastropods, and bivalves as well. So all of them have specific adaptations to such extremely low oxygen in their environment. Okay, so typically an example of an animal's adaptation to OMZs is that they have high concentration of hemoglobins, so large respiratory features in their bodies, but with very small thin sizes. Also, they have a high concentration of pyruvate, oxidorhizomyces. So this one is something like a catalyst enzyme present in a bacteria or archaea uh, that are present in these types of environments. And also, the organisms that live here manifest the presence of sulfide oxidizing symbionts. Okay, the last heterotrophic ecosystem for this particular topic is the abyssal plain. So, abyssal plain is the largest ecosystem in the planet, and of course in the deep sea environment. And as you can see in the picture on the right, the abyssal plain, those light green colored portion here on the picture. So this part here at the bottom illustration, but looking at the comparison size of the area around here or above it with the adjacent habitats like seamounts or canyons or continental margins. So the abyssal plains are actually very, very large in size. So the abyssal plain lie beyond the continental slopes at a depth between 3,000 to 4 or to 6,000 meters. And one of the typical features of the substrate of the abyssal plain, is that they covered with a very thick layer of sediment. Usually, the abyssal plain, as you can see on the picture at the bottom, right? So the clays are accumulated by thousands of meters in thickness. So and that's stretched across thousands of kilometers across the seafloors and create a monotonous seafloors. It looks like a boring one. So the temperature in the abyssal plain is at the average of 2 degrees Celsius, and the average salinity is 35 parts per thousands. Okay, so mostly the waters here are saturated with dissolved oxygen, um, but a very low concentration of oxygen. And probably the absence of light is one of the important factors that affect the organism that lives in the abyssal plain. Okay, so despite the fact that the abyssal plains are so deep that we might thought that there is actually no such environmental disturbances in this type of environment. So actually there is a physical disturbances in these areas. And probably the most notable examples are garbage pollutions that reach the bottom of the oceans. And of course the deep sea mining that extract minerals from the bottom of the ocean. So probably the main reason why the abyssal plain conservation is needed uh, is because the abyssal plain supports a very high biodiversity that is composed of the diverse macro and meiofauna like nematodes and foraminiferans. So the foods here are very limited, and the organisms must be really good at conserving energy and also good at looking for food as well through adaptations. So in the picture here on the right, for example, um, muddy abyssal seafloor in the California coast, so just below 4,000 meters. As you can see, those littered silvery stuff at the bottom here are example of dead bodies of salps, and that provides a food for many organisms that live in there. Okay, so large megafauna in the abyssal plains are mainly composed of holotorgans, or the group of sea cucumber, echinoderms as well, decapod crustaceans, and of course the fish. And finally, the sessile fauna are mainly composed of crinoids, sponges, and entozoans that are found on the hard and soft substratum of the abyssal plain. Okay, so moving into the chemosynthetic ecosystem in the deep sea. So this environment, or these habitats are simply driven by chemicals, and the marine organism that thrives here are actually taking advantage of these chemicals in order to survive. So probably the most popular chemosynthetic ecosystems in the deep sea environment is the hydrothermal vents, or the geothermal vents. So how are they formed and where are they located? So they actually located, or they are the mid ridges are actually a chain of underwater volcanoes. So this is an area where two tectonic plates are being pulled together. So they are being pulled together apart from each other. So um, when the split of up to two tectonic plates happen, so cracks are formed in the crust, and the cold seawater with an average temperature of 2 degrees Celsius seeps through these cracks. So when the cold water penetrates through these cracks in the ocean, the superheated fluid that comes from the seafloor push back the cold water. And so when they mix with the uh, water following the um, precipitation in the water, that could create a dense black smoke that rises from the hydrothermal vents. Okay, so today the mid-ocean ridge system has 7 hydrothermal vent biogeographic provinces, and as you can see here in the picture on the right, that their respective location in the globes are mapped. Okay, so the first hydrothermal vent was discovered in 1977 in the East Pacific mid-oceanic ridge, and the scientists named these vents as the black smokes. So they found out that these vents emit geothermally heated water with an extremely hot temperature of about 400 degrees Celsius. So this superheated fluids emits high level of sulfides that precipitates when contact with the water in the ocean. So this is actually form a mountain of precipitated chemicals on the seafloor of the vents. So as you can see in the photo on the right, so this shows a black smoke that spills from the tectonic crack, and this one is from the mid-Atlantic ridges. In 2000, they discovered this new type of alkaline deep sea hydrothermal vent that is located just off the axis from the mid-ocean ridges. And then this new type of geothermal vent was named as the Lost City, and that was discovered on the seafloor of Atlantis Massive Mountain in the mid-Atlantic ridges. So the photo here at the bottom is actually a 5-foot white ledge in the chimney in the Lost City. So those white stuff that you can see here are actually what they call the dendritic carbonate, I think. Yeah, it's a dendritic carbonate growth. So this actually form when fluids that are rich in minerals seeps through the ledge and comes in contact with the cold water. All right, so the hydrothermal vents are classified either a black smoker, or a white smoker. So both of these types of vents are rich in minerals such as like calcium, iron, potassium or carbon dioxide, hydrogen sulfide, and so on and so forth. As you can see, uh, those chemicals from the picture here at the bottom, that these rich minerals come from the interior of the seafloor and push up to the surface of the water by many tectonic activities or tectonic cracks that splits on the seafloor. So one of the most abundant and popular organisms that try the thermal vents are the tube worms. So these tube worms are very big in size and they have no mouth and digestive system. However, they have a specialized adaptation by the presence of the tryposom. So a tryposom is basically a sac inside the body of the tube worm, and this tryposom is filled with chemoautotrophic bacteria. All right, so a tube worm or the tube worms take oxygen, carbon dioxide, and hydrogen sulfide in the hydrothermal fluid and store them in their tryposom. Specifically, these chemicals were stored in the blood vessel of the tube worm around the tryposom. And then the bacteria synthesize these organic chemicals to become useful for the tube worms. And yeah, that is in the form of organic matter then. So this picture here at the bottom right corner shows a community of tube worms that grows in the lava near the hydrothermal vent in California. So since their discovery in 1977, around 590 species that comes from the hydrothermal vents have been already described by science. And out of 600 discovered species, about 400 of these have been identified as endemic to the hydrothermal vents. All right, so the major faunal groups that are found in these areas are the tube worms, of course, the bathymogillid mussels, the clams, the shrimps. As you can see here in the photos, sitting around one of the vents in these areas. And finally, the crabs, amphipods, and the polychaetes also thrives in here. Okay, so when the hydrothermal vent was discovered in 1977, the scientists found a single-celled bacterium that has no chlorophyll that can be able to produce sugar. Also, what they found around that vent was that the water are rich in hydrogen sulfide chemicals, and for most of the living things on Earth, so this molecule is actually toxic, but not for this type of bacteria in the vents. So the vent bacteria the vent bacteria actually adapted to use the energy that comes from the hydrogen sulfide molecules. So rather than doing the photosynthesis, so because obviously there is no sunlight in there, the bacteria conducts a chemosynthesis instead. So the process of chemosynthesis involves the utilization of 6 carbon dioxide plus 6 dissolved oxygen from the water and 24 hydrogen sulfide molecules. And by doing the chemosynthetic process, so these compounds are converted into glucose or sugar, sulfur, and water. So this bacteria use this byproduct for the chemosynthesis to, of course, survive and let other animals to survive as well. So the organism depends on the chemosynthetic bacteria as their source of food. All right, in 1993, rather, so scientists are able to identify more than 100 gastropod species in the hydrothermal vents. For example, the Pompeii worm in the photo here can be able to tolerate the temperature of about or up to 80 degrees Celsius. So this Pompeii worm was discovered in 1980s. Also, another type of gastropod that thrives the hydrothermal vents is the scaly-foot Rhesomalon species. See here. So in this picture here. So this particular gastropod uses the iron sulfide to build its dermal sclerites as an alternative to calcium carbonate. So instead of using calcium carbonates to harden the shells, because as we know, calcium carbonates dissolve so easily in the deep sea because of the high concentration of the carbon dioxide in the water. So this process, or this species, uses pyrite and greigite in the iron sulfide instead to harden the shell. And that's one way of how organism in the deep sea adapts to this hostile environment. All right, so the tube worms in the vents are actually a great example of an animal-bacterial symbiosis in the vents, and many great examples are still existing. So because the temperature in the vents reaches up to 80 degrees Celsius, so scientists classify the bacteria that live in there as thermophiles. However, for the vents areas that have a temperature of up to 350 degrees Celsius, the bacteria are then classified as hyperthermophile. So the thermophile have a very simple diet, which is based on the metals or gases and minerals that present in the area. So today, marine scientists are able to actually produce the bacteria that they collected from the vents. So by means of culturing them in the lab setting and feeding the bacteria with only sulfur, oxygen, and carbon dioxide. So that's the symbiont, which is the bacteria, converts inorganic molecules in the form of oxygen, and carbon dioxide, and hydrogen sulfide to become organic molecules that the host animal uses as a source of nutrition. So in 1993, marine biologists were also able to confirm that endosymbiosis exists within the clam gill tissue. So a type of these clams are shown in the picture below. So this in 1984, the hydrothermal vent mussels and vesicomid clams also carry endosymbionts. Actually, before the discoveries in 1983 and 1984, it was in 1981 that the scientists were able to know that the bacteria and the tube worm symbiosis actually exist. So from the picture above, on the right, the body of the tube worm has no digestive system. But instead, the tube worm replaces it with the troposome. So in the black colored portion here in the image are actually the bacteria that thrives or that live in the troposome of the tube worm. So the bacteria utilize the minerals from the water through the chemosynthesis and feeds the tube worm with the byproducts, organic molecules that comes from the said process. And then in return, the tube worm gives also the protection for the bacteria. Okay, so not all host animals have endosymbionts in their body because some have episymbionts, or the symbiont or the bacteria in particular live on the animal externally as opposed to the inside of the animal. So shrimps at the vents, for example, in the mid-Atlantic ridge were once thought to have been not benefited from the symbiosis of for the macroinvertebrate survival at the vents. But that changed in 1988 when the scientists discovered an episymbiosis in the hydrothermal vents. Such example of this is in the Lepidothryllis phocensis, like the one in the picture, that that has the episymbiosis relationship. Okay, so the hydrothermal vent is not the only ecosystem in the deep sea that is driven by chemicals or chemosynthesis, because in fact the cold seps is another chemosynthetic environment in the deep sea. So cold seps, or sometimes also called as cold vents, are actually areas on the seafloor that are located or that are loaded with energy-rich chemicals such as methane or hydrogen sulfide and other hydrocarbon-rich fluid compounds that seeps through or sifts from the ocean floor. So normally this is in the form of a brine pool. So as you can see in the photo on the right, the yellow arrow here um, indicates the passage of chemicals, of all the chemicals, all the way to the surface of the water through seepage. And that could, in the cold seps, it's basically easy to identify by having the bubbles that sip from the seafloor. So this normally these are the methanes that sips from the seafloor. Okay, so cold seps were discovered in 1983 at approximately 500 meters depth around the western Florida in Gulf of Mexico. And cold steps communities are more stable than hydrothermal vents. So, by looking at the geographical activities that happen in this two chemosynthetic ecosystem, of course, the hydrothermal vents will likely to change faster than cold steps due to the volcanic activities that happen in the hydrothermal vents. And for that reason as well, the hydrothermal vents are short-lived, whereas the cold steps are long-lasting deep sea communities. Okay, so in the picture here, this is a cold step area around the offshore Virginia. You can see those bubbles that comes out from the seafloor. So these are actually methane bubbles, and chemosynthetic bacteria are able to use such compounds to make food for the organisms. Cold steps are actually we may also find oil and gas in the cold steps. Or we can also find brine steps that form brine pools like the one in the photo. So this actually what I called it the brine pool. Okay, a cold step is a biome that has a higher number of animals and plant endemism. So over time, they develop unique topography by forming carbonate rock and reefs. And most of the organisms that live in there are actually dependent on the bacterial activity. So, for example, an, an icate, a hydrous calcium carbonate compound can be associated with oxidizing methane or on a cold seps. There are two types of cold seps, and that is based on their location in terms of depth. So the first one is the shallow cold seps, and the other one is the deep cold seps. The organisms that live in the cold seps are called extremophiles, since they can be able to tolerate such extreme condition in the area like super acidic water. Uh, Yes, because of the high concentration of methane in the cold seps. So this compound is actually oxidize the carbon dioxide and leads to the to the changes in the chemistry of the water, which makes the water in the cold seps acidic. Um, so the microbial communities in the cold seps are the basis of life in there because they are the one that take advantage of the deep sea energy source in the cold seps. They are actually aggregate in the microbial mats. Um, and they actually metabolize the methane and hydrogen sulfide, a source of energy for many marine organisms in there. So this is basically a process of chemosynthesis that these bacteria are doing specifically in the cold seps. This is pretty much different. This is ultimately different from the chemosynthetic activity that happens in the hydrothermal vents. So today, unlike the hydrothermal vents, the cold steps are least explored with very few potential locations that are to be explored. And since its discovery in 1993, only about 230 species have been described so far by science. And one of the most common organisms that describe and live in the cold steppes are the sulfide mining shoeworms, like the one in the photo on the right. And they can actually live for over 250 years, according to some marine scientists. All right, so in particular, the methanotrophic archaea are the one responsible for the anaerobic oxidation of methane in the cold steppes. And they use the process for their growth, and they use this to for the growth and reproduction, and the reproduction of the important organic compounds such as the lipids and sugar. So the carbon dioxide in the water is the main source of carbon for the compounds that they make. So the photo here on the right is a methanotrophic bacteria that are found in the cold steppes. And these bacteria feed on the methane and hydrogen sulfide and convert the energy source to become bicarbonate hydrogen sulfide plus plus water. Okay, so just like in hydrothermal vents, in order for this type of ecosystem to thrive and sustain, so a symbiotic relationship must occur, must happen. And by doing the chemosynthesis, the bacteria supply the energy needs by many organisms in order to survive. And in return, the organisms that have symbiont bacteria in their bodies provide shelter and protection. So a typical food chain in the or a typical consumers in the community can be illustrated by the bacteria as the base or the first trophic level in the food chain. Okay, so the the bacteriivores, or those animals that feed on the bacteria like snails, worms, and crabs, are the typical primary consumers of bacteria in the cold steppes. And then shrimps and some crabs, like those in the picture on the right, actually feed on the detritus organic matter produced by mussels, clams, and shoeworms in the area. And then macro and megafauna species such as octopus, fish, and crustacean are attracted to the community to complete the food chain. All right, so the other reducing habitat under the chemosynthetic ecosystem in the deep sea environment is the whale falls. So when a whale died, it goes down to the bottom of the oceans and become the living community that is driven by chemosynthetic activities in that area. So in 1987, so Craig Smith, when he was boarding the submersible Alvin in the North Pacific, accidentally crossed with a whale skeleton on the seafloor and found out that the chemosynthetic community is thriving there. And not just whale that becomes a community when they died and falls in the bottom of the ocean, because sunken wood, kelp, and other organic matters also advance rapidly as a chemosynthetic community in the deep sea. In the case of whale falls, it has three steps in the ecological progression. So the first one is the scavenger phase. So during this stage, the flesh of the whale is, of course, being eaten until what's left are the bones and and they are exposed in the on the ocean floors. And then the second phase follows. So what they call it the opportunistic phase. So at this stage, the exposed bones and the sediments are being colonized by the opportunistic folicates and crustaceans. And then the final phase, which is the chematotropic or sopophilic phase, complete the ecological progression of the whale falls community. So at this final stage, the bones of the whales are actually composed of 60% lipids. And then the bacteria are then degrading these lipids to produce sulfides that are being used by chematotropic microorganisms that allows them to subsequently colonize the area. Okay, so the topic 6 is about the ecological patterns and processing in influencing the structure of the deep sea assemblages. And one of the first of this pattern and process that I'm going to tackle a bit is the distribution. So according to one theory, or the Vicariance theory, the plate tectonic activities has a major role in the distribution of the marine organisms. So for example, in Australia, at about 90 million years ago, so Australia drifts close to the South Pole, but every year it moves a few millimeters away from the South Pole and drifts closer to the equator. And that actually influenced the distribution and dispersal of the marine organisms in that particular biogeographic region. So how is that happening? For example, since Australia was once closer to South Pole, so the water was then very cold, and probably after a million of years ago, so the water becomes hotter and hotter as it gets closer to the equator. So the animals that are not actually capable to quickly adapt to that changing temperature environment. So the result is they may tend to change their distribution pattern just to find the perfect or suitable habitat for their survival. So they just change location to feed and to reproduce, and of course to live. Okay, so according to many marine scientists, the importance of the dispersal varies with local oceanographic features. So, for example, the direction and intensity of the currents, as well as the biology of such organisms in that area. So that could influence the distribution patterns. And another influencer of the dispersal or distribution of marine organisms, as well as their extension, is of course the intervention of humans. So for example, pollution has great effect on the distribution pattern of marine organisms. So of course, if the water is polluted in that habitat, so they tend to find, or the organism that live in there may tend to find another home, and that changes the ecological pattern of their distribution, because the once fish that live in there, or could be could be not in there anymore because of the pollution that actually disturb their surrounding and feeding patterns. Also, mining, fishing, and there's very typical disturbances like recreational activities in the shallow waters, like in the beach. So all this actually either directly or indirectly could have an influence in the ecological patterns, and rather the distribution of the marine organisms in the deep sea environment. Okay, so about 90% of the organic carbon are exported or respired within the mesopelagic zones, and that produces about 30% of the biological carbon dioxide production in the ocean. So the microbial respiration rate in deep sea from 150 to 1,000 meters are of the orders of magnitude of 3 to 4. So for the microbial abundance and biomass in the deep sea environment, so that the prokaryotic Zooplankton and micronecton biomass declines exponentially with depth, so meaning that from the euphotic to the bathypelagic zone, the biomass for these groups of microbes decreases by the magnitude of 2 to 4 orders. But the microbial diversity for some species shows increase with the depth. So, for example, in Radiolaria, the diversity often increases as we go deeper into the abyss. And finally, the microbial biomass is greater in higher latitude and decreases towards the trophic regions. Okay, so that means the microbial biomass in temperate regions around the mesopelagic zone are higher in temperate regions in comparison with tropical regions that or where the sea surface temperature is hotter. Zooplankton and nekton in the deep sea. So the vertical migration of zooplankton results in the peaks in the abundance and biomass of many taxa in the mesopelagic zone during the night. So, for example, in one study about the ostracods in the North Pacific region, so scientists found that ostracods were actually peak in the mesopelagic zone during the night and becomes lesser in abundance during the day. So basically, they are absent in the upper 50 meters during the day and becomes too many at night due to their vertical migration activity. So another example is the ontogenetic vertical migration of the copepod species that results in seasonal mesopelagic peaks in the abundance of the copepod during the night. Okay, so the topic about the fish ecology in the deep sea is actually a very broad topic about the ecology of the deep sea community, but I choose to include this subtopic here just to give you a bit of information about this interesting component of the deep sea ecology. So the mesopelagic zone is a home to a significant portion of the total fish biomass on Earth. So the mesopelagic fishes are found almost around all the oceans in the Earth, so except for the ocean in the Arctic. So in 1980, it was estimated that the biomass, or the fish biomass, in the mesopelagic zone reached 1 billion tons. And then in 2008, it reached up to 2 billion tons in biomass. So the picture on the right side of your screen are the lantern fishes. So they are one of the most abundant fishes on the mesopelagic zone. Sorry for that. So the mesofish is represented by 35 families, and they are dominated by Mectophidae with 245 species. So again, the picture on the right is in fact Mectopidae, which is represented with 35 genera. 35 genera. Okay, so until just recently, so the bristlemouth in this photo here at the bottom becomes the most abundant fish in the ocean with hundreds of trillions or quadrillions of tons of biomass was recorded. Many mesopelagic deep sea fishes have swim bladders that help them to control buoyancy. And as we know that this is very important because if the fish does actively swim all the time, so the fish will likely to use so much energy. And because of the presence of swim bladders for these marine fishes, so they become hard to study because of the chambers in their bodies that contain gas, and that could easily burst out when they come up from the water. But today, scientists have already developed a way to actually catch this fishes alive, and bringing them up to the surface of the water using the such sophisticated machine and technology. And of course, they control the pressure in there. So for to depressurize the fish, it comes from the very intense pressure at the bottom of the oceans. The faunal composition of the Venta community. So the deep sea habitats of the upper bathyal zone from 200 to 2,000 meters deep. So these areas are around these dips are mainly characterized by the dominance of hard bottoms, especially around the seamounts. So the major meiofauna species here are the foraminiferans and nematodes. And the macrofauna are dominated by polychaetes, like the one in the photo on the right, and paracarid crustaceans and bivalves. So the major components of the megafauna in the benthic community are the predatory brittle stars, so large scavenging fishes, and the amphipods as well. So the biomass and density of the benthic organisms rapidly decreases with distance from land. And recent discoveries in the benthic communities in the deep sea are showing that lineages that were known from the fossil records were actually found alive in the benthic community. So particularly in those benthic organisms that is flashed in the bottom right corner of your screen. So these are the organisms that were discovered in New Caledonia exploration. And the scientists thought that these species are already extinct, but not anymore since they have already found this in the wild. Okay, so benthic community in the deep sea is indeed a very challenging seafloor to live, but all the benthic fauna that live in here have learned to evolve to such extreme pressure, cold temperature, and even the scarcity of food. So similar with the other type of ecosystem in the deep sea, so the photosynthesis is not possible in the benthic community because there is no light to support the said process. And also, the extremely cold water temperature slows down the growth of bacteria and other microorganisms that live in there. So mostly, the microbial community in the benthic deep sea environment is sustained by the rich chemicals or minerals in organic matter that comes from the detritus. So for example, animal faces and the decaying tissue or dead bodies of large animals or large mammals like whales, especially around near the surface of the water. So the benthic communities in the mesopelagic zones as well as in the deeper region, the abyssal and the hadal zones receive organic matter from the turbidity current that delivers rich organic matters. So occasionally, food may arrive in the benthic community in the form of the carcasses of whales, for example, and other large marine organisms like shark, which basically sinks faster all the way to the bottom of the ocean. So what are the type of foods found in the benthic community, and what is the typical benthic food chains looks like? So foods in the benthic community may come in form of allochthonous material. So this means that the foods may come from elsewhere, except that there's no photosynthesis involved. So basically, that comes in the benthic community or in a form of organic matters and nutrients, usually nitrogen, or not usually those organic matters have nitrogen and phosphorus compounds. However, some particles like marine snow and fecal pellets are not directly being eaten by the benthic organisms. Instead, the bacteria decompose them first before the primary consumers of the bacteria eat them. And as we have already discussed, similar with the other deep sea environments, so the food in the benthic community are patchy and limiting, and that actually restrict the size of the body of the creatures that live in there, as well as their abundance in the area. So about 20% of the total organic production that comes from the epipelagic zones make it to the benthic community in the mesopelagic zones, and only about 5% of what's left for the abyssal in Hadal benthic community. So that's why food is so much limited in benthic communities in the very deep abyss in comparison with the benthic communities about around the mesopelagic zone. Okay, so another form of how food arrived in the benthic community is in the form of autochthonous material that comes from the hydrothermal event. So this is the result of a chemosynthetic activities by the bacteria that live in there. The meiofauna composition here are the foraminiferans, so nematodes and small forms that feed on bacteria and the sediment. And of course, the first trophic level, the primary producers of the benthic community are, of course, the bacteria, and the foraminiferans, nematode, small worms feed on that bacteria. So the infauna worms and bivalves then feed on the meiofauna. So they are the secondary consumers in the typical food chains. However, about 80% of the secondary consumers are deposit feeders such as sea cucumber or brittle stars and sea urchins. And in the benthic community, only less than 7% of the species that live in there are suspension feeders. However, the top predators in this community are classified as fishes, squids, and of course sea stars. It was actually a long misconception before that when we say about the deep sea, so the first thing that will comes in our mind when we heard the deep sea is yes, it is a lifeless and boring landscape at the very bottom of the ocean. But the truth is, deep sea holds a mega diversity of marine organisms, and and as much as an ordinary person question this sort of misconceptualized idea, so the scientific community actually held few hypotheses to break this kind of belief or thinking. So in 1967, there was a paper that was published in a scientific community, and that fundamentally shifted the long-held belief of many of the deep sea, many that the deep sea is a lifeless environment in the abyss. Because prior to 1976, so many had thought that the extreme environmental condition in the deep sea is devoid of life and, biologically speaking, a desolate wasteland. And then in the year 1967, so Hessler and Sanders, with the use of their newly developed sampling device, so they found out that the deep sea is in fact a place of megadiversity hotspot at the bottom of the oceans. Then, after that, they began to throw theories about how this very extreme and unforgiving type of environment able to support a mega diversity of marine organisms in there. So what Sanders did was in 1968 he took a paper and put it on his whiteboard, and then he wrote there the word specializations, and he suggested that because the deep sea is monotonous and predictable, meaning that the landscape at the bottom of the ocean is pretty much constant, and that doesn't change that much. So the population evolved over time until they become specialized in how they feed. So Sanders called this theory as the stability time hypothesis. But people's curiosity didn't stop there, because in 1972, Robert Dighton and Hessler, this time, scratched the word specialization in Sanders' stability time hypothesis and wrote the word predation and biological cropping on the whiteboard instead. So basically, that's what they were trying to prove was feeding in the deep seas, either intentionally or unintentionally. So, meaning animals in the deep sea can eat other animals either by purpose, like hunting their prey, or unintentionally, like it just happened that one animal come across into stuffing into his mouth, and then suddenly um, food comes in to their mouth without the animal's intention to actually feed that. So this cropping, or what they called biological cropping, whether accidental or not, so that reduces the competitions and eventually preventing one or a few species to monopolize the resources in the deep sea. So Dayton and Hessler call this theory a biological cropping hypothesis. And then aside from these two hypotheses, many other ideas comes next. So today, the terms predations, disturbance, or pathogens, and successional dynamics were just a few of the terms that were added on a list of hypothetical questions that are trying to answer the curiosity of the many on how or why the deep sea organisms are so diverse in nature. Um, so for you, which would you think of these two hypotheses best suit to answer this type of question? All right, so many of the deep sea organisms are gigantic in size and many are also smaller in size. But both of them can live up over decades or centuries. So, for example, the round-nosed grenadier, based on a study of their otoliths, can live up for 60 years. Sablefish can be able to live up to more than a century, and the rough-eye fish may live for more than 2 centuries. So, for mollusk, like clams, can also live for more than 100 years. The question is how they manage to do that. So, the simple answer are because that from natural causes in deep sea environment is typically low, and the growth is often very slow for many animals that live in there. So, of course, because there is less predators that forage in this very uh, uh, deep part of the oceans. Another explanation is that the reproductive age of the deep sea animals is often later than of those animals or organisms that live in the shallow area, so particularly in the euphotic zones. So, for example, the roundnose grenadier again, so that elongated fish in the photo on the right. So this type of deep sea fish began reproducing itself after 14 to 16 years, and for many other fishes in deep sea, this could be actually much more longer than that. Also, for the tripod fish and lizard fish, so it is very rare to encounter their potential mates. But in order to sustain their population, so they adapted this kind of condition where individual develop both males and females organ at the same time. So that actually allows an individual to do a self fertilization. Perhaps the most common way on how to find mate in the deep sea is to use a unique kind of light patterns. In fact, anglerfish uses such kind of light pattern on its tail as well as using scents to find potential mates. And once a male anglerfish found its female counterpart, then the male uh, to attach himself to the male or the female using his hook teeth. So this is pretty much like a parasitic relationship, but actually it's not a parasitic relationship, but more on a type of symbiosis among the two um, uh, individuals of the same species. So as you can see in the photo here, so the male attached himself to the female, and once he's okay and comfortable on his position, then he starts to merge his blood vessels with the female. And then in exchange for giving blood of the male, then the male or the male provides a very reliable sperm sperm source. And as I have said earlier, the deep sea animals grow to maturity very slowly. So because they also have a very late reproductive age. So if, for example, the fish is overfished, then the population may take decades to recover naturally. So as one of the great examples of the fish population that are likely being affected by the overfishing is the orange roughy. The sterling photo on the right one, on the right, the bottom right corner. Okay, for the last topic in this report, I will give a quick run about how our organisms survive in this unforgiving deep sea environment. So the first subtopic about the survival of the organism in the deep sea is on how this organism adapt to intense pressure in the water. So most of the deep sea environment is under the pressure of about 200 to more than 1,000 atmosphere. So it's so intense that uh, if a person tried to dive in such dip, so his skull will likely or quickly crush into pieces. Actually, if you remember in the topic one when I discussed about the historical exploration of Picard in the Challenger Deep in the 1960s. So originally, their plan was to make an observation of the Challenger Deep for hours, but due to the pressure in that deep, so the window of their submersible vehicle cracked, and that made them decide to explore the dip for only 20 minutes instead of staying there for hours. So surprisingly, echinoderms and mollusks, despite all their small body sizes, are able to tolerate the high pressure in the deep sea. So they managed to do that uh, by tolerating such high pressure because this organism maintain fluid pressure within their body tissues, and that corresponds exactly to the pressure in their surrounding environment. So physiologically speaking, the tissues fluid inside the body of this organism pushes against the surrounding pressure, and then they do that with the equal force but on opposite direction. So by doing that, this prevents them or prevents the organism from being crushed in the uh, deep sea environment. Okay, so another effect of pressure is that as the pressure increases and the temperature decreases, so the calcium carbonate, which is the main compound needed to make a hard shell of bone, so the solubility of calcium carbonate increases as well. So meaning, as the animals go deeper and deeper into the ocean, so these animals experience difficulties in creating shells out of carbon carbonate. So therefore, they tend to either have no shells or have thin shells, or make shells out of other mineral found in this area. So for example, as I have said earlier, the sulfur iron can be used by some gastropod as an alternative to calcium carbonate. Okay, so the protein in animals' body are affected by increasing hydrostatic pressure of the deep sea environment as well. So this creature must learn how to actually modify the proteins and membranes in order to survive such intense pressure. So the deep sea fishes have actin protein uh, in their body. So, but what exactly is this actin? So the actin is actually a type of protein that is very important for different cellular functions. So, for example, the actin serves as a main component for muscle fiber. And then, in order for an organism or a fish to survive in such intense pressure, so the fish must change the mechanism on how to process the actin protein in their body. So, um, one adaptation for this particular scenario is that some species change the active sites of actin in their bodies, and that also changes the salt bridge pattern of the protein in their body. So when they do this, they have more advantage or power rather to stabilize the ATP binding, ATP binding in their bodies. So eventually that helps them to survive such intense pressure. So based on the study, they found that the deep sea fishes have more salt bridges in their proteins, so particularly in their actins. And in comparison with the fishes that inhabit the upper zone of the sea, um, one of the examples of deep sea that undergoes this type of adaptation is that the Coryphenoides armatus fish, this one in the upper right corner of your screen. Okay, so specific osmolytes are abundant in the deep sea fishes, but what exactly are osmolytes? So osmolytes are actually compounds in the body of a fish, and its primary importance is to protect the cells from being drying out, meaning it maintains the fluid integrity of the cells in the body of the fishes. Okay, so scientists found out that as they go deeper, so that the trimethylamine nitrogen oxide is in fishes also increases, and high TM the high trimethylamine nitrogen oxide replaces the other osmolytes in the body of the fishes. So what exactly is a trimethylamine nitrogen oxide? So the main role of this is to prevent the distortion and compression of proteins in other important molecules in the fish body. So because they don't do that, with so much external pressure, so that the fish proteins and other vital molecules will be distorted and compressed and will cause a catastrophic consequence for a fish, which is the death. So for example, this abyssal grenadier fish on the top right photo on your screen has so much trimethylamine nitrogen oxide in this body, and that protects the proteins that inside the body of the fish from high pressure by adjusting the osmolytes in its body. So another example of amazing adaptation of fish in the deep sea is that of the Mariana Hadolf snailfish. So they are the only known fish that inhabit the deepest Mariana Trench so far. Okay, so they develop a specialized modification in the osteocalcin gene, so meaning they regulate bone development and doing some sort of frameshift mutations, which resulted in an open skull and cartilaginous bones in their body. So if you are a fish and have skulls, or have skull and hard bones, so when you go deeper as the Mariana Trench, your bone will crush due to pressure. But this species of snailfish is able to withstand such pressure because of that kind of adaptations or makeshift mutations. Eventually, the snailfish will be able to survive the pressure in the shallower water when we try to bring it up the water surface. Okay, some fishes have swim bladders during their juvenile states because they tend to inhabit the upper pelagic zone to feed and grow into maturity. However, when they become adult, so they must return to the dark abyss, and as we know, the food in the dark abyss is totally scarce. So therefore, in order to save energy and slow down their metabolism, so they either wither or fill their swim bladders with fats. And finally, for some bacteria, they undergo homoviscous adaptations, or they construct membranes from more fluid. So literally, it was like constructing more walls to protect yourself from intense pressure or something. Okay, so how organisms survive or adapt to extreme temperature in the deep sea? So in the deep sea, the temperature is constant. There's no seasonal or annual variation in the deeper portion of the at more than 3,000 to 4,000 meter deep. So the temperature is between 1 to 0 or 1 to 4 degrees Celsius. However, there are a few exceptions because the Mediterranean Sea has a temperature of 13 degrees Celsius, and the and about 20% or 20 degrees average temperature for the Red Sea. So basically, animals that live in these two geographic regions must also adapt to this average temperatures. So perhaps the most common adaptation to cold environment is to, of course, slow down your metabolism. And by doing that, so organism must move slowly, and grow slowly, and even reproduce less frequently. However, cold temperature brings an advantage for the organisms living there because they actually require less food to take, or uh, they may live longer in comparison with the animals in the photic zones. And also, they may grow larger in size. Okay, so the density increases as we go deeper, and there is not much less energy to take in order for an organism to stay afloat in the water. And because of that, most of the fishes, if not all, don't have the swim bladders. So because the density is very similar to their surrounding environment, so meaning they don't need to take extra effort to swim in order to prevent themselves from sinking at the bottom. Okay, adaptation to the scarcity of food in the deep sea. For many organisms in the deep sea, again, metabolism requires only a less amount of oxygen in the water, and by doing that, they can live for a very long period of time without consuming foods. So typically, the food in the deep sea community comes from the uh, organic material that falls from the above uh, the surface of the water. So, for example, dead bodies of large creatures like whale and shark, and of course from chemosynthetic activities uh, by the bacteria. The ossification adaptation is common for deep sea vertebrates because they have less muscles and less ossified bones. So, like the gopher fish in the photo on the right, so due to the scarcity of food, animals or fishes specifically tend to have huge mouth, and back pointing teeth for better grasp of this of their prey. And also, they have specialized bioluminescence light either on their tail or in their head or around their bodies. So here is a trivia questions. So why do deep sea spider crabs go so big? So despite the fact that there are not so many food at the bottom of the oceans. So the first is we need to understand that the cold water holds oxygen that crabs needs, but also they have very slow metabolism to process all the oxygens they consume. And then they utilize that energy from the body or from the food they take, but because they consume they consume is much oxygen in the water, so their cells do not need much of this oxygen to metabolize at some stage. And once their body gets rich in oxygen with slow metabolic rate, and where their cells don't need so much of this oxygen, so the wasting of the oxygen in the spider system or the crabs, the system of the crab isn't possible. So, but what's possible is that it creates more and more new cells for the body of the crabs. And having more new cells means more tissue for the crabs. Therefore, more tissue means the body gets bigger and bigger. And this simple explanation is simply applies for the bigger animals that live in there. Okay, so adaptation of the deep sea animals in the low level of oxygen in the water. Some organisms have hemoglobin in their body, and this is very efficient in the RMZs. So for example, we have here the giant red mysid, which have highly developed gills and thick blood to water diffusion distance that actually enables the species to effectively remove as much as 90% of the oxygen in the water. So some also have hemocyanin in their body. So hemocyanin is a type of protein that binds so easily with oxygen. And also some use nitrate instead of oxygen through the process of denitrification. Okay, so to adapt to the extreme darkness of the deep sea, so many organisms develop a bioluminescence adaptation. So bioluminescence occurs through a chemical reactions. Usually this is in the form of luciferin molecule. So some animals or organism also speeds up the reaction of the luciferin to produce a quick bioluminescence reactions, and by doing that they actually use the catalyst called luciferase. So these organisms are able to control the light they can produce by regulating their body chemistry and the brain processes. So for example, this lanternfish here on the right side has light producing photophores on his head, which acts like the flashlight. So some also bind the luciferin with oxygen, so they call it the photoprotein. So it is like a bioluminescence bomb. So because when a certain ion, for example, when a calcium ion becomes present in the water, so the bioluminescence will automatically light up like a bomb. Some also produce red bioluminescence, like the dragonfish in the photo here on the right, the bottom right. And by doing that, they can be able to see red-colored prey under the darkness of the deep sea. Okay, so for the last word, the bioluminescence has extraordinary function for the organism that have it in order for them to survive at the very extreme darkness of the deep sea environment. So for example, they can scare or confuse the predators using the bioluminescence, or use a counter illumination or something like a camouflage technique to hide themselves from their prey, from their predators. And also they use it to find mates and for communication as well. And finally, to attract or deter their prey. Okay, so I hope you find this topic interesting and made you some and will make you more interested to reread the files or the print PDF form of PDF format that I'm going to send through the Google to you in a Google class. So please have a look at that and have a time to study it. And thank you for listening on my very long